Hello friends, you're watching Edurythms Biology, where we add rhythm to your biology. Yes friends, your biology. If you're preparing for CSIR-NET-JRF in life sciences, or you are preparing for MSc entrance in biological sciences, or you are preparing for a medical examination NEET, or even if you are a basic entry-level student of biology, then this video is for you. I'm your host and friend Dr. Zubair Shanib, and today we're going to talk about enzymes, an overview. So let's begin. When you look at the enzymes, before we begin, I just want to give you an analogy of what enzymes are actually and how do they work. So consider that you are going to college and you find that there is strike, and then you decide to go walking to your college. Once you have reached your college, the first class is already over. You are sitting in the next class now, and you are thinking about the third class. Should I attend or not? Because you know there is no way back, or again you have to go walking. So what is happening is that you are being slowed down. You are doing your job perfectly, but you are slowing down and you are also missing your classes. So that is a disadvantage. Now what you do regularly is, in the morning you wake up, you take a ride, either a bus, a scooty or a car. You take anything, you reach your college on time, you attend the first class, you attend the second class, even you attend the extra classes. Why? Because you know there's a ferry back home. So that applies to the chemical reactions as well. Let me give an example of a chemical reaction. So let us say that we have hydrogen peroxide. This hydrogen peroxide can break down into water and oxygen. Fine. So you have this reaction in place. If you allow it to go off its own, it will take either months or years for the hydrogen peroxide to break down into water and oxygen. But what if you provide it a ferry? What if you provide it a ride? So you provide something called as catalyst. The term is catalyst. Catalyst. So what does the catalyst actually do? The catalyst speeds up the reaction from, say, hours to years and down to the seconds. Fine. One of the fastest known enzyme known to mankind is carbonic anhydrase. Carbonic anhydrase. Carbonic anhydrase, as you know, in case of carbon transport, carbon dioxide combines with water to form carbonic acid, a bicarbonate, H2CO3. H2CO3 bicarbonate. The enzyme involved is carbonic anhydrase. Okay? The enzyme involved is carbonic anhydrase. You can imagine that it catalyzes around 36 million molecules of, it hydrates around 36 million molecules of carbon dioxide per minute. And if you break it down, it means roughly around 500,000 molecules of carbon dioxide per second. Fine. So this is what enzymes do. This is what the catalyst does. It speeds up the reaction without itself being used up in the reaction. Now I give you another example. The same concept applies in case of cells. Fine. So carbonic anhydrase is present in your red blood cells. Fine. So it is present in the red blood cells. Now here we do not call them as catalysts. We name them as enzymes. Fine. So what are enzymes? They are actually biocatalysts made up of a biomolecule. For example, I will let you know what it is made up of. For example, proteins. Okay, this being done, I have given you the analogy. Now we will talk about the discovery. So before I venture into the discovery, what does the enzyme actually do? Does it initiate a reaction? And the answer is no. Does it speed up the reaction? The answer is yes. Does it provide energy for the reaction? The answer is no. So what does it actually do? What enzymes do is they lower the activation energy for a particular reaction. And now you will ask, what is activation energy? Let me take you again to a next example. Consider that you have to cross a mountain. Okay, so you are right now here, you have to cross a mountain and then you will reach some place. Fine. So let us say this is a reactant A and this is the product B. Fine. This is your graph. Fine. So you are right now here. So what you have to do is you have to climb this steep slope and then reach here, and then you can roll down here and reach here. Okay. So let us say this energy is the activation energy, EA. So a reaction cannot occur, or it takes a lot of time to reach its activation energy. So it will occur very slowly. But what enzymes offer it offers them a simple tunnel. So if I take a tunnel out of the mountain, so now that reactant A will pass through the tunnel and it will be easy for him. So what does actually enzymes do? They lower the activation energy needed for a particular chemical reaction. Fine. So it provides an easy way out. Now this is the activation energy in the presence of enzyme. Okay. So you can see it's lower than the actual energy. Fine. So this is how your enzymes, you catalyze your reactions. Now let us go forward and talk about its discovery. The discovery of enzymes is very unique. In what way is it unique? Let me discuss it. So it was Louis Pasteur, who you well know for its pasteurization of milk. He gave a concept that yeast cells are able to ferment glucose into alcohol, that is ethanol. That's what he said. But it was way back in 1897 when Edward Buchner was working with the yeast cells. And while working, the yeast got spoiled. So he tried to add glucose to it. And to his surprise, this glucose converted into ethanol. So he called it as in yeast. Inside yeast, there is something that is converting the glucose into ethanol. Fine. So he's added an extra step to the Louis Pasteur. He said it's not the living cells that are required for the fermentation, but it is inside the cell any kind of liquid that is converting this. So what was that liquid? That liquid was named as enzyme. En means inside, and zyme stands for ZYME. Zyme stands for yeast. So he named it as inside yeast. So enzyme. So you can say that the enzyme that was present inside the yeast, it is not the cell that's converting this, it is the enzyme, something inside the yeast cells that is converting it, and thus came the name enzyme, came into existence. So he said that that enzyme was later named as zymase. So what does zymase actually do? First, it uses glucose. As you know, glucose breaks down into pyruvate. So it takes up pyruvate, and converts this pyruvate first into acetaldehyde, CH3, C double bond OH, and then it's being converted into ethanol, that is C2H5OH. This happens in the yeast. The process is itself called as fermentation, okay, and it releases carbon dioxide. Okay, so this is the reaction, the ENZYMES catalysis, and this was a little bit about its discovery. Fine. So now let us move on to the third point of enzymes. So by the time we have now known about the analogy, discovery, now we will talk about the principle of catalysis. So what's the principle of catalysis? The principle of catalysis is a reactant A combines with a reactant B, and with the help of the enzyme, the reaction takes place. Okay, so let me give you an idea about it. How does it happen? So I will give you an idea of the principle of catalysis by the enzymes was first given by Fischer. So Fischer said, so he said that consider your enzymes as a lock. Okay, so here we have an enzyme. It looks like a lock with a hole in it. Okay, and now it says further that now consider your substrate as a key. So here is your substrate and this is the key. So what the enzyme has to do is, enzymes either break the bonds, or they form the bonds, or they transfer some group from one form to another, or they transfer electrons from one group to another, or they use water to break something, or they join something. So these are the different functions of the enzymes. But actually, how does it do it? So let us say that this is the enzyme, okay, and this is the substrate. So Fischer said that the substrate has the enzyme has something called as active site. Okay. Now this active site is actually, it is a three-dimensional. So enzyme is actually a three-dimensional entity which with an active site. It's a hole in the lock and it's surrounded by the amino acid sequence, maintained by not only the sequence but also by the configuration of the amino acids, whether it's an aromatic, whether it is hydroxylated iron. For example, you have serine proteases. They have serine on the hydroxyl group on the active sites. Best example is chymotrypsin. Okay, come back to the concept now. So the substrate binds, the key enters the lock and it breaks the bond here. Fine. The bond is broken. Now it forms something called as enzyme substrate complex when the key is inside the lock, and then it converts into the enzyme product. So what happens is when the key is inserted, it starts changing into something else. Now the key looks like this, and this. Now what we get out is we have this triangle out, and then we have this is being converted into dome shaped structure. So now we have dome or semicircle. So we get the products. Fine. So this is the principle of catalysis, that enzymes combine with a substrate at an active site. The reaction takes place. Koshland said that induced fit theory. It is not the lock and key only theory, but the key has the ability to change the configuration of the enzymes. Fine. So both the concepts are actually true. The lock and key theory also, and the Koshland's induced fit theory is also true. So when the induced fit theory takes place, now the substrate is changing into, it's breaking the bonds, making the bonds, and now it's changing into a product which is completely different from the substrate. If you remember the substrate as a key, it was like this, a round head with a rectangular tail. Now it has changed into product. The rectangle has converted into semicircle, and the knob, the circle, has converted into product. What I mean is the substrate is no more there. Now we have the products. So this is the principle of catalysis. Now once we have talked about the principle of catalysis, let us now talk about activation energy. So as I started with my lecture, that what enzymes actually do is they decrease the activation energy of a reaction. So I have already talked about this. Now let us talk about the chemical nature of the enzymes. Point number 5. So what are actually enzymes made up of? So scientists came up and they said that enzymes are made up of proteins. Okay, agreed. Enzymes are made up of proteins. Fine. So let us say that we have a three-dimensional enzyme. Okay, and this is its active site. Fine. I will represent my active site with a different color so that you can understand it. So this is your active site here. Okay. So the protein part of the enzyme, is called apoenzyme. Apoenzyme. So apoenzyme will not be functional until it gains its other non-protein parts. So what are the other non-protein parts so that it will become a complete enzyme? Okay, let us talk about it. So now there are two kinds of non-protein parts attached to the enzymes. One is your prosthetic group. Prosthetic group. The prosthetic group is bound tightly to the enzyme by a covalent bond. Fine. Now we also have something attached to the enzyme called as cofactors. Cofactors. Now what are cofactors? Cofactors are which are loosely bound to the enzyme. It means they are important for the function of the enzyme. Once the job is done, they can leave the enzyme. So there is no need of them. So what are cofactors? Cofactors, because they are loosely bound to an enzyme by non-covalent bonds. Fine. So we have two kinds of cofactors. One is organic cofactor. Organic cofactor. And another is inorganic. Inorganic. So what are organic cofactors? Let us now talk about the organic cofactors. The organic cofactors are generally called as coenzymes. Coenzymes. So you can see they are a part of the enzyme. Co means a part of the enzyme. They are part of coenzyme which helps them to function properly. Okay. This being said, now what are coenzymes? Coenzymes have been found that they are often vitamins. They are often vitamins. And inorganic ions, for example, magnesium is a good example. Then ferrous is also a good example of the inorganic ions. So what you are seeing here is a protein has an enzyme has a protein part called apoenzyme, and then it also needs non-protein part. If it's tightly bound to it, we call it as a prosthetic group, and if it's loosely bound to it, we call it as a cofactor. Cofactor now can be organic or inorganic. Organics are called coenzymes, and coenzymes are often vitamins. And inorganic ions like magnesium is a part of DNA polymerase. Fine, if you remember DNA replication. And iron is a part of catalase, which has started begin my lecture with hydrogen peroxide. Catalase breaks into water and oxygen. Now talking about the prosthetic groups. Prosthetic groups, best example of prosthetic groups is when you talk about iron porphyrin ring in cytochrome C oxidase. Cytochrome C oxidase. Okay, it's completely bind to the cytochrome C oxidase. It doesn't leave it anytime. Now, when you talk about now this coenzymes, I have two examples for you. One is vitamins, because they are often vitamins. One is B2. B2 is riboflavin, and from the riboflavin vitamin we have something called as FAD, that is flavin adenine dinucleotide. It's a coenzyme. I can give an example of this. If you remember Krebs cycle, you have succinate being converted into fumarate, and there you find an enzyme called as succinate dehydrogenase, and there you can FADH2. So it's a coenzyme. Another example of coenzyme is vitamin B3. It's called nicotinamide or niacin, and it forms what is called as NAD. NAD positive. So this is nicotinamide adenine dinucleotide. Fine. Nicotinamide adenine dinucleotide. What's the best example? Best example is when you convert malate into oxaloacetate. There you have NAD. So both are 4 carbons. Malate converts into oxaloacetate, and there NAD accepts and becomes NADH2, which later becomes the source of ATP synthesis in the Krebs cycle. Okay. Now this being said, now one thing you have to remember is that enzyme is complete now of both proteins and non-protein parts. So now you have a different term for the enzyme. You call it now as holoenzyme. Holoenzyme. So holoenzyme is, what is holoenzyme? It's an apoenzyme, a protein part, and a non-protein part. So this is complete holoenzyme. Now when I talk about holoenzymes, what do I mean by holoenzyme? Okay, a protein part and a non-protein part. It's understood. Got it. Now, what do you mean by this holoenzyme? You might have seen in transcription. In transcription, you have an enzyme called as RNA polymerase holoenzyme. Holoenzyme. So where do you find this enzyme? You find it in E. coli. It's a prokaryotic cell. It is made up of alpha 2, beta, beta dash, and sigma 70. So when you talk about holoenzyme, it means it is made up of a protein as well as the non-protein part. Accepted. No confusion right here. Now when we talk about RNA polymerase holoenzyme, we have to understand this enzyme is not made up of one polypeptide. It's made up of more than one polypeptide. And whenever a protein is made up of more than one polypeptide, we call it as subunits. Good. So we call it as subunits. Subunits. Okay, we call it as subunits. Now how do subunits look like? Okay. So let us see that we have the alpha 2 subunits, then the beta subunit, then the beta dash subunit, and then the delta subunit. Okay. So this is alpha, beta, beta dash, and sigma 70. Now you can see that there are many subunits in an enzyme. And now we have to talk about the domain. Let me give you a concept of the domain. Wherever you find the active site for the binding, because RNA polymerase has to bind to the DNA, so it has to have an active site where it binds to the DNA. So let us say that this active site is in the beta subunit. Okay, so let me put it like this. We have here the active site. We have here the active site. So the subunit which contains the active site, this subunit is called domain. So domain is a part of a subunit where you have the active site. Again, I will repeat it. So if an enzyme is a holoenzyme, now we have different subunits. Any subunit which contains the active site is called a domain. Fine. So you can find that it is the beta subunit where the domain of the enzyme lies. This is just an example I'm giving you. Okay. Now you have understood the chemical nature of the enzymes. Now let us move further, and we have also talked about the active site, the three-dimensional sites, and now we'll come to the classification of the enzymes. So friends, you might have noted that if there is zymase, so it ends with ase. Fine. So you will also find many enzymes with the classical names like pepsin, trypsin, chymotrypsin. Okay, these are classical names and they have still stuck to this name. Now when we add ase to the enzymes, it gives an idea that it is an enzyme. So but how do we nomenclature them? How do we nomenclature them? First we talk about nomenclature and then the classification. So how will we nomenclature them? It's very easy. We can nomenclature them by using the word ase. You can use the substrate. For example, glucose converts into glucose 6-phosphate. We call it as glucokinase. So glucose is the substrate and kinase is the activity of the enzyme. So you can name the enzymes on the basis of their substrate and their activity. Similarly, we have isomerases. that is phosphotriose isomerase. What does it do in glycolysis? It converts your dihydroxyacetone phosphate into glyceraldehyde 3-phosphate or back and forth, because it's an equilibrium reaction. Fine. So this is the name, how we name the enzymes. Similarly, now when we classify them based on the function they do. So let me give you an mnemonics so that again you can remember the enzyme classification. Okay, let's go. So I will give you a mnemonics. It will help you to remember it very easily. So we have something called as OTHILL. O-T-H-I-L-L. O stands for oxidoreductases. Oxidoreductase. Okay. T stands for transferases. Transferase. H stands for hydrolysis. Hydrolase. I stands for isomerase. L stands for lyase. And another N stands for ligase. Let me give you one example of each. Oxidoreduction, when your malate changes into oxaloacetate, so that is oxidized and the electrons and the proton are taken by NADH2. That's an example of oxidation reduction reaction in your Krebs cycle. Talking about transferases, the best example is hexokinase. What does it do? It converts glucose into glucose 6-phosphate. So what is it transferring? It's transferring phosphate from the ATP onto the glucose and making it glucose 6-phosphate. Yes, first reaction of the glycolysis. Talking about hydrolysis, when we eat proteins, we need water to digest them. Right? For example, pepsin needs water to digest the proteins. So when proteins are being degraded with the help of water, we call this kind of reaction as hydrolysis. Now we talk about isomerase. The best example I already gave is the glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate or vice versa. The enzyme involved is triose phosphate isomerase, again a reaction of glycolysis. Okay, now we talk about Liasons. When something is broken without the help of water, the example is called Liasons. Okay, for example, I will give an example. When you, again the glycolysis example. So you have the fructose 1,6-biphosphate. With the help of lyase, it breaks into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So that's an example of lyase. Now talking about the enzyme called as ligase, you might have heard about in DNA replication, the end enzyme is ligase. So what does it do? It joins the Okazaki fragments by using the hydroxyl group on one side and the phosphate on the other, forming something called as phosphodiester bond. Okay, and this requires energy in the form of NADH2 in case of E. coli and energy in the form of ATP in case of the eukaryotic cells. So friends, this is what enzymes are all about. Only I have given the summary. So in the next lecture, we will be talking about it in more details like enzyme kinetics, enzyme inhibition. So please keep tuned and do subscribe my channel by pressing the bell icons. And if you have any comments, you can give the comments below. I will respond to them as soon as I see them. So friends, how we summarize them? The enzymes are very specific. Enzymes need cofactors to work with them. They catalyze a specific kind of reaction. Fine. And they can be denatured. They have an optimum pH to work with. They have an optimum temperature to work with. If you increase the temperature, being a protein, it will denature. Okay. And you know that enzymes have a very high molecular weight. For example, urease has a molecular weight of 400. Okay. So this was just a scratch on the enzymes. We will take it in more detail in the next lectures. Thank you for watching my channel. Thank you and best of luck. Bye bye.